Hello, class. I was reviewing the instructions for doing the dispersion calculation for the oak trees or any other species that you would like to do. And I had you measuring the distance between individuals so that you could use that in a calculation. And that's one way to do this sort of analysis from field data. But then I realized that there might be an easier way using the iNaturalist website and an Excel spreadsheet. So I wanted to show you how to do that in case you wanted to do it. And you can, by the way, use this for your stomata dispersion as well. So let me share my screen with you and we'll go through some screens that will help you, I hope, to do this. So we'll start with a quick iNaturalist thing. I want to find my species, Quercus agrifolia. Okay, Coast Live Oak. And then we'll do Lower Arroyo Park, Pasadena. So now there we are. I can do remove the bounding box and I can see all the oak trees in Lower Arroyo Park. So then if I pick an area, so let me just take this so I can make this simpler, and I can zoom in a little bit on an area. I want to zoom in so I can see the separated individual observations, but not so much that I can't see more than, say 10 observations. In fact, this has several hundred on it. So this makes a good zoom in distribution that I can then analyze. And so to analyze this, I'm just going to take a screenshot and I want to capture it so that I have at least, one observation. So I don't go way over here or something, maybe bound it, try to make it reasonably square or whatever, something like that. So boom, I take a screen cap. So that's all we need this screen for. So let's get rid of that. Then I import that screen cap, the JPEG, into a Word doc. So here it is in a Word doc, and I put a grid over it. And if that's a little difficult to imagine, here's a Word doc. If you start at home, let's see, let me go in order here. If you go to View, you can tell it you want to see the ruler. And that gives me the ruler at the top and the side that I can use to make my grid. So I set that up initially, and then I go to draw. No, not draw, sorry. Insert, insert shapes. I pick a line, and now I can set a line. So then I go on my rulers in both directions. I already set one here, but I could do another one. Let's kind of zoom in a little bit, so I have the edges a little easier to see. So now I want to set a line from here. I go to the first 1 inch mark and I run it across. And then let's see, just cut and paste, grab another one, grab this line, nope, and move it down. To the next inch line. And then, I can click on, I can say, oh, I'd like to insert another line. I don't want to bore you with this, so I'm not going to build the whole thing. But I go out to the next inch, and these are approximate, right? And I'm hoping to get them about straight, but I'm making a grid, right? And into this grid, I can import a photo. So if I go find my screenshot photo there, grab it, I can stick it right in here. I can set it up like this. So I just grabbed that photo, stuck it in. If I need to change the dimensions a little bit, I can grab a corner to make it bigger or smaller to fit on the page, right? Anyways, once I go through this process, I'm gonna get rid of this, don't save it, go away. I end up with a grid like this. And this is where I can begin my analysis. So let me go to the instructions from that link that I gave you. So this was the instructions from the citable link for calculating dispersion. And it says in there, so I broke down the paragraph that was on that link. And the first one is to make a grid, right? You have a set of quadrats. Each one of these quadrats, these squares, is one sample. And then it says that that what we want to do is count how many individuals are in each square and keep track of them separately. So they did one where they had samples, and they counted, like there's 25 in one square, 25 in another square, 25 in another square. And then there's a bunch of squares with 0, 0, 0 in it, right? So that's the first step. Okay, so first step, count the number of individuals in each square. So what I did was I set up a spreadsheet, right? And each square in this, I have 6x5 grid, right? So 6 times 5 is 30. I have 30 squares, and I just count. So like in the first square, there's 1. In the second square, I count 26. In the next square, I count 13. And I just keep doing that for the whole set until I have all 30 numbers. Some of the squares have 0, but I just put 0, right? So that's step 1. So let's go back to those instructions. 1,000 files open. So the first one was counting the squares, done. Take the mean of that. Oh yes, that's done too. Right down here, we have total observations, and I just divide that by 30, and I get 6.5. That's the number I want. Oh, look, it's almost the same as their number. And that you want from the first step. Second step is to calculate the variance. Now, variance is a descriptive statistic that tells us how variable the data is, like where is the variation. To do that, you take the number in any square, subtract the mean, and square it. So I'm going to do that 30 times, right? 30 times. I'm going to take the number, subtract the mean, square the number. So you can see, let me click on this, that I've programmed this cell in Excel to do that, right? B2 is this square minus 6.53, which is the mean, times itself, gives me that number. And so going back to our instructions, we do that for every single cell. And right, and then we add them all up, get the sum of those variances. And so in this case, for theirs, it was 125. I do the same thing here, add them all up. Here's the total. The total variance is 2195. Yeah, a big number, much bigger than their number. Okay, so that's the process to generate the number we're going to interpret. I'll tell you how to interpret it in just a second. But it has a couple steps. You import a photo from iNaturalist at the right scale so you can see the separate observations, but not too separate. You import that into Word and you make a grid over that. And then you use that grid to count the number of individuals in each square. Yeah. Putting the data into a spreadsheet like this will allow you to calculate it equally or easily. Here's the other sort of secret to this then, is that the interpreting of it, when you get the variance, is to see, is it less than 1, which would mean that the distribution or the dispersion is uniform, meaning there's approximate equal distances between the individuals, or if it equals 1, they're random, yeah, randomly distributed. And if it's greater than 1, which ours is, it's clumped. And when you find individuals clumped in a population, it means that either the conditions that they need to grow are found only in certain places and then they all live there, or that they work together and form a unit. So like animals that flock or herd would be found in this situation. Plants that form a unit, like a forest, that might interact in some way, tend to join together and form in a clumped sitting. So from my quick analysis, I can see that the oaks in this area are in fact clumped at this level. So pretty easy. Nice for interpretation and whatnot. keep in mind, once you've built this grid in Excel, that you can take the photo out and put in another photo. You can just keep doing that. Yeah. So once you kind of play with this, you set up a Word doc, you can import other photos into that same grid and use it over and over and over again. And the scale doesn't really matter that much. So if I had a photo of a, I don't know, a leaf that I had the stomata on, I could drop that right in here and do the same analysis and find out if they're clumped or regular or uniform, whatever it is. Yeah. So so that's it. I'm going to post this Excel spreadsheet on the Canvas page. So you're welcome just to grab it and use it if you want, or you can kind of program your own if you feel like it. But that's essentially it. That's all there is to doing this. And you don't have to use the data that we collected in the field distance between individuals, because then you'd have to do a lot. This is so much easier, and you can do it sitting at home or, you know, out in your shed, just like I am. Anyways, so thanks for listening. I hope that works out, and I hope to see you soon. Adios.